This diagram tracks four distinct chemical molecules, an ergoline in LSD, two tryptamines in psilocin and DMT, and a phenylethylamine in mescaline. Despite diverse structures, all four converge precisely on a single GQ protein-coupled receptor known as 5-HT2A. In 1998, Franz Vollenweider's lab and later Katrin Preller's team tested the absolute limits of this convergence. They administered a drug called ketanserin to human volunteers directly prior to a dose of psilocybin or LSD. Ketanserin is a highly selective antagonist. It slides directly into the 5-HT2A pocket and sits there, occupying the site without activating the receptor. This image captures the control room during the scanner-based trials. With the receptor physically blocked by ketanserin, the psychedelic remained fully circulating in the bloodstream, yet volunteers experienced zero subjective effects, and the large-scale cortical connectivity changes normally induced by the drugs completely vanished. The physical presence of the psychedelic in the body yields nothing if this specific receptor is occupied. The empirical data confirms that 5-HT2A is the definitive biological gate for the entire psychedelic experience. Humans produce a natural ligand for this receptor. Serotonin binds to these very same 5-HT2A targets continuously from birth. Under normal physiological conditions, your own serotonin uses this receptor to tune baseline cognition, regulating daily attention, mood, and basic cognitive flexibility. A direct functional comparison reveals a discrepancy. Identical high-affinity binding to the same receptor by endogenous serotonin does not trigger ego dissolution, nor does it induce the sudden, rapid growth of new dendritic spines. The receptor is the same, the chemical binding is the same, yet the divergent output suggests that our model of how this receptor translates chemical contact into a biological cascade is missing a variable. Profound neuroplasticity and altered states of consciousness depend on more than just binding. The difference lies in lipid solubility, cellular anatomy, and access to a hidden intracellular signaling network. To locate that missing variable, we must map where these receptors are clustered at the macroanatomical level. This medical illustration details layer 5 pyramidal neurons located deep within the prefrontal cortex. The 5-HT2A receptors are expressed at extreme densities, precisely along the apical dendrites of these specific cells. These large, deep-layer neurons act as the brain's primary association hubs. They integrate massive streams of information and carry the high-level predictive coding priors that we use to model reality. When a psychedelic activates 5-HT2A on these exact neurons, the immediate result is a localized surge of cortical excitation. The cells trigger an asynchronous release of glutamate, dissolving the tidy, synchronized rhythms of standard cortical activity. This desynchronization disrupts the rigid flow of top-down predictions. Bottom-up sensory data flows with far less filtering, establishing the biological foundation for the relaxed beliefs under psychedelics, or rebus model. That explains the shift in consciousness, but it does not explain the timeline. If the molecule simply docks and fires, the resulting disruption should be brief, yet an LSD state routinely lasts for 10 to 12 hours. This animated model visualizes Brian Roth's cryo-EM imaging of the 5-HT2A receptor at the atomic scale. Notice the structure hovering directly above the bound LSD molecule. That is the extracellular loop 2, or EL2. When LSD enters the pocket, the EL2 folds directly over it, like a physical lid. It traps the compound inside, reducing its dissociation rate to a near halt. The intensity of the state is governed by the location of the receptors on layer 5 neurons, while the duration is dictated by the atomic architecture of a single extracellular loop. The physical trap of the EL2 explains why the trip lasts so long, but it leaves the core biological discrepancy unresolved. A trapped molecule on the cell surface still does not explain why psychedelics trigger physical neuronal growth while serotonin does not. In 2023, David Olson's lab at UC Davis isolated the answer. It resides entirely in the chemical properties of solubility. This cross-section of a neuronal membrane illustrates the limitation of endogenous neurotransmitters. Serotonin is hydrophilic, meaning it loves water and cannot penetrate fats. When it approaches the lipid bilayer, it must rebound entirely, restricting its access to 5-HT2A receptors on the outer surface. Classic psychedelics are lipophilic. Because they are highly fat-soluble, they slip straight through the lipid membrane and flood into the internal cytoplasm of the neuron. Once inside, they reach a separate, hidden population of 5-HT2A receptors. These receptors are anchored directly to the surface of the Golgi apparatus. Under standard biological constraints, serotonin is physically barred from this intracellular address. Activating the same receptor produces different downstream effects depending on its location. Surface receptors generate electrical excitation, while intracellular Golgi receptors trigger physical rewiring. Docking at the Golgi apparatus ignites a specific intracellular signaling cascade. It forces the expression of BDNF, which binds to TRK-B, culminating in the activation of the mTOR growth pathway. Activating the mTOR cascade drives a rapid physical consequence. The neuron actively sprouts, branches, and extends new dendritic spines, increasing synaptic connection density. The neuroplastic power of these compounds follows a distinct route. Activating the intracellular pool on the Golgi apparatus is what triggers the structural remodeling. Mapping the cellular address of neuroplasticity presents a clear pharmacological target. The current focus is to engineer compounds that decouple structural rewiring from the subjective hallucination. This effort has produced a class of molecules called psychoplastogens. A psychoplastogen is defined by its ability to rapidly promote structural neuroplasticity regardless of subjective effects. Tabernanthalol, or TBG, is a leading candidate in this class. It is an engineered, non-hallucinogenic analog derived from the chemical structure of ibogaine. In animal models, TBG penetrates the cell to trigger the mTOR pathway and retain neuroplastic benefits, but it does so without driving the severe layer 5 cortical desynchronization that causes a trip. Pharmacologists are achieving this precision by exploiting a phenomenon known as biased agonism. This block diagram maps the internal logic of biased agonism. A single activated receptor splits its signaling down diverging internal routes, typically the GQ versus the beta-arrestin pathway. Certain molecules bind to the receptor in a way that leans the output heavily in one direction. By reverse engineering the 5-HT2A receptor's atomic structure, researchers are building new molecules to exploit this bias. The goal is to maximize the intracellular growth cascades while actively starving the surface-level hallucinogenic pathways. We now have the atomic blueprints of the receptor and a map of its internal forks. Modern drug design is actively attempting to chemically navigate those internal forks to isolate the clinical medicine from the subjective experience. The 5-HT2A receptor is the prerequisite for the classic psychedelic experience. When activated on the surface of layer 5 neurons, it disrupts predictive coding, floods the cortex with glutamate, and temporarily dissolves the boundaries of normal consciousness. When activated on the interior of the cell at the Golgi apparatus, it engages ancient growth cascades, physically expanding the dendritic architecture of the brain. That cellular expansion is the mechanism required to overwrite pathology. It forces open a biological window where the rigid, entrenched neural circuits underlying severe depression, addiction, and trauma can be physically remodeled. Deciphering the exact molecular kinematics of this single receptor takes the scientific field beyond merely observing altered states of consciousness. It provides the biological schematics required to physically repair the human mind.